So we're starting the questions now. Part C. Describe, using information from your graph, so we're going to have to use the graph, how the change in concentration of pectinase affected the mass of extracted apple juice. So there's 2 marks available here, and so it's the basic trend and then anything in particular about the trend. Is it linear or not? Does it level off? That sort of thing. So we need to use the names of our independent and dependent variables. So we're going to, as concentration of pectinase increases, the mean mass of juice also increases. So you've got that positive correlation. So as the concentration of pectinase increases, so does the mass of juice. So that's our basic trend. And then we need to say whether the relationship is, proportional or not. So ours, it's not directly proportional because you can see the line starts to level off or plateau. So the relationship is not always proportional, as the line starts to level off, which is our plateau. So eventually this will actually level off completely. Identify, using information from your graph, the optimum concentration of pectinase for extracting apple juice. A bit of a strange one, that optimum. Well, we'd expect an optimum with a pH experiment or a temperature experiment, but not particularly with concentration. So it's our best concentration really, so it's got to be 2, hasn't it? It's got to be 2 there, so 2 gives our best mass of juice. So 2%, 2% and the units there already. Give one reason why the pectinase concentration you identified is the optimum. Well, why are we choosing 2? Because it gives the best mass of juice produced. So it gives the most mass of juice. So this gives the most mass of juice produced. Give one reason why it was important to stir. So we stirred our, there's my experiment, we stirred the apple and we put in the pectinase. So if we just put the pectinase in, it'd probably sit on the top or sink to the bottom, but it wouldn't be mixed up. So we need to mix the pectinase with the apple. Let's get rid of those graphs. So it evenly distributes the substrate and the enzyme. You have to mix and evenly distribute the substrate, which is the apple, and the enzyme, which is the pectinase. Then we have E. Describe how you made sure that the mass of extracted apple was measured correctly. So this is really what you did to make sure you maybe start thinking about controlled variables between the different treatments. So things like, did you filter these for the same amount of time, or did you filter them? Did it tell you how to filter them? Let's have a look. We filtered it, and it says after 5 minutes. So filtered for the same time, 5 minutes. Filtered for the same time, which was 5 minutes. So that's one of our marks. And then really, what else did we do with recording to make sure it was accurate? Well, when you get a balance, then you put your sample on the balance. Then this surface has got to be flat, and there's not got to be any kind of drafts or anything like that. So we can mention something about the balance. So, balance on a flat surface. What else do we need to do with this balance? It will sometimes, before you put anything on, it'll be already reading something. So there'll be a little button with T on for tare, which will zero the balance. So reset or tare the balance. When we're recording this information, we could make sure that we're recording to the same decimal places each time. So record to same decimal places each time. Sometimes bits of apple or whatever might get spilled on this balance, so remember to clean it. So clean the balance each time. So that's the mass, and there are a number of other possibilities for this experiment, but we need 3, so 3 of those will do. And then part F is really thinking about other things that affect enzymes. So this is, one of your controlled variables was temperature. So do you know a bit of enzyme theory about temperature? So pectinase has an optimum temperature, then we've got the classic, below the optimum and above the optimum. So if we were drawing a graph of this temperature and the rate of enzyme activity, it'd be our classic kind of graph there. So this question is talking about this bit there, and then that's that bit, and then this question down here is talking about that part of the graph there. So below the optimum, what would happen? How would that affect the rate? Well, the rate would be lower. So this is our optimum. So the rate would be lower. Or it would take longer to extract the juice, that sort of explanation. Why? We just need some classic bit of enzyme theory, level 3 science, biology. Then less kinetic energy collisions have lower energy, less enzyme substrate complexes formed. So a bit of biology theory there, and then above the optimum, what's going on there? Again, just more classic biology. So enzyme rate would be lower. So again, the rate would be lower. We can talk about denaturing of the enzyme. Active site loses its shape. Substrate no longer fits into active site, so can no longer bind. So classic biology there. And that's the end of question 1.